From Diamond Light Source, this is the Diamond Podcast.
Welcome to the Diamond Light Source Podcast with me, Meera Senthilingam. This month, we're moving out to the clinic to see how Diamond's beam lines are being used by clinicians nationwide. We'll be finding out how X-rays are being used to identify the potential risks of metal-on-metal hip replacements, as well as for finding biological markers for faster diagnosis of cancer.
And then we can see differences maybe in the fat content or in the proteins or in the DNA. And we're using these differences, which we call markers, for in the future to diagnose cancer at single cell level.
Josep Soler-Suso will be explaining how his team are looking into finding these biomarkers in lung cancer cells later in the show, when we also investigate the causes of preterm labour.
Most women deliver at term, in other words 40 weeks of pregnancy around that time, but about 10% of women deliver less than 37 weeks, and that does seem to be on the increase, and we really know very little about why that happens.
And Anna David will be giving us an insight into how the analysis of amniotic membranes could maybe one day help us predict who is likely to go into preterm labour. So all that insight into the clinic, plus the latest news and events from Diamond, coming up in this June edition of the Diamond Light Source Podcast.
The Diamond Podcast. For more information, look us up online at diamond.ac.uk/podcast.
The users of Diamond Light Source come from a wide range of scientific disciplines. One recent discipline is medicine, where clinicians are using Diamond's X-rays on samples donated by patients in order to research a variety of medical problems. This could offer great contributions to future medical advances. So this month's podcast is focusing on the range of clinical research taking place using the synchrotron. The first being the work of obstetrician Dr. Anna David from University College London. Anna is trying to find out more about the causes of preterm labour by studying the structures of amniotic membranes, the thin wall of the amniotic sac in which fetuses develop in the womb. I went along to University College Hospital in London, where Anna is based, to find out more about how she's looking into this.
Most women deliver at term, in other words 40 weeks of pregnancy around that time, but about 10% of women deliver less than 37 weeks, and that does seem to be on the increase, and we really know very little about why that happens. The problems with babies that deliver preterm is that many of them who deliver very early end up having problems with brain development, lung diseases, and we know that later on in life they end up having cardiovascular disease and hypertension. So there's a big research effort to try to find out why women deliver preterm.
How common are preterm labours? So how often do they happen?
So preterm labours probably happen in about 10% of women during pregnancy. We know that about 4 out of 10 cases, the membranes surrounding the baby rupture early, and many of those women go into labour early, fairly soon after the membranes rupture.
What are thought to be the main causes then?
Well, for some women there does appear to be infection. We know that women who develop an infection get inflammation of the membranes are more likely to rupture their membranes. There are other reasons, for instance, women who might have heavy bleeding in early pregnancy. The blood appears to weaken the membranes in some way and causes them to rupture early. And lastly, stretching the membranes, for instance, in twins or triplet pregnancies, or in women who have extra fluid around the baby, something called polyhydramnios, seems to put extra pressure on the membranes and causes them to rupture preterm.
So now, in order to understand why this happens, you've set about looking at the actual structure of the amniotic membrane and looking into what changes in the structure to cause these ruptures.
Yes, so the amniotic membrane is a sort of bag that carries the baby. The baby is surrounded by fluid, and then there is a sac around the baby. And usually the placenta is near the top of the uterus, and then the membranes are around the baby and down near the bottom of the uterus, down near the cervix or the neck of the womb. Now, there are actually 2 layers to this sac. There's the amnion, which is the internal membrane, and this is a single layer of epithelial cells. And there's the chorion, which is is the outer layer of membrane. Now, the amnion is made up of collagen fibres, and it's the collagen fibres that seem to bring the tensile strength to the membranes, because the amnion is actually incredibly strong. Now, we know during pregnancy, at the end of pregnancy, the amnion alters its structure in the place down near the cervix. There is a zone of altered morphology down near the cervix, which is the area where the membranes are much more likely to rupture.
Now, to help set the scene a little bit about just how this membrane is structured, we've got a placenta here in front of us. So tell me more about this placenta. It's a little bit bigger, actually, than I thought it might be.
Well, it is quite a big thing. A placenta is really an amazing structure. It feeds the baby for 40 weeks, and then suddenly around the time of the end of pregnancy, you go into labour and the placenta is delivered. As you can see, there are two layers to the membrane. The inner layer is really very fine. We can peel off the outer chorion, and the inner layer is really very fine. And if I push my finger through the membrane, you really have to push it through quite a lot, and it still hasn't ruptured. And you can imagine that this baby is surrounded by sometimes up to a couple of litres of fluid, and yet it still doesn't rupture.
It's actually incredibly strong, and it's very thin, so it's basically transparent, extremely thin, and I'm pushing my finger right through it now, and there is no sign of that bursting or giving way at all.
If you stretch the membrane, actually the tensile strength of the membrane, the amnion membrane, increases until it reaches a threshold above which it ruptures. So it is a very clever piece of biological machinery which prevents us going into labour before we should do.
It seems here then that the amniotic membrane really can take a lot of stress. So how are you actually setting about looking into the structure of this and trying to understand what really happened in order for this to burst early?
Well, we're looking at the major component of the amnion, which is the collagen fibres. Nobody's really looked at how the collagen fibres function, how they're spaced out, how they're aligned, how dense they are in the membranes, and then how this might correlate to why the membranes rupture.
Now, which beamline are you using at Diamond, and how does this enable you to actually visualise then the structure of the collagen within these membranes?
Well, we're using the beamline called I-22, which is the non-crystalline diffraction beamline. The X-rays go through the amniotic membrane. We line up the amnion in the X-ray beam. And what happens is, as the X-rays hit the collagen fibres in the amnion, some of them are scattered by those collagen fibres. And we can work out by the amount of scatter, the alignment of the collagen fibres, how closely spaced they are, and how many they are in the membrane.
So what have you been able to visualise so far? So how is the collagen structured in this particular area?
Well, we found that the collagen is actually structured in quite an ordered fashion. So we've looked initially at term membranes from women who've delivered on time. What we found is that there's a difference in the way the collagen is aligned in membranes that are sampled from near the zone of altered morphology, near the neck of the womb where it ruptures, and membranes that are further apart. So in the membranes near the cervix, the collagen is more lined up, the fibrils are more lined up, and they're also slightly more spaced apart in comparison to membranes that are near the placenta. And you might hypothesise that that would actually increase the risk of the membranes rupturing in that area.
And have you been able to look at samples of membranes from people that have ruptured preterm?
We've put the membranes in the beamline, and we found that in comparison to the term membranes, the collagen fibres are more lined up. So we think that there may be some difference in the preterm compared to the term membranes.
Are there any thoughts as to what this predisposition to this could be, or kind of what the causes of this could be, or how could you essentially hopefully use this to know who might therefore rupture early in the future?
Well, we're basically looking at how the different factors that we know increase the chance of your rupturing membranes early, how they affect the collagen structure in the amniotic membrane. So we're going to be doing some experiments in the next beamline by incubating the membranes overnight with blood or with bacterial products like lipopolysaccharide that are related to infection that perhaps might break down the collagen, or actually repetitively stretching the membranes and then seeing how those factors alter the collagen structure in the amnion. And then we may be able to say more about really why those factors affect the structure. The other thing is that we know that there are genetic predispositions to rupturing membranes early, and it may be that these are related to the collagen structure, but obviously a lot more work is needed on that, and really the synchrotron facility is the only place that we can do this kind of work.
So lots more to find out in order to be able to predict who may be prone to preterm labour. That was Anna David from University College London. Now, moving away from the womb and on to diseases that affect you later on in life, one of which is cancer. There are many forms of cancer affecting millions of people globally each year. Many of them can be treated if caught early enough. So any diagnostic method that could help identify cancerous cells in the early stages could make a dramatic difference. Dr. Josep Soler Suso is an oncologist at the University of Kiel, and he's using Diamond's infrared beamline to study lung cancer cells in hope of finding markers that could help spot cancerous cells early on in their tracks. I spoke to Josep to discover how he's doing this.
What we're trying to do is we're using the Diamond synchrotron, the beamline B22, to study single cells. Some of them are lung cancer, some of them are normal cells. And the fact to use the synchrotron is because it allows us to look inside the cell, smaller areas in the cell, and then we can see differences maybe in the fat content or in the proteins or in the DNA. And we're using these differences, which we call markers, for in the future to diagnose cancer at single cell level.
Now, how do you set about using infrared? So how does it enable you to see more about what's going on inside the cell?
Infrared, especially using a synchrotron, allows me to look different parts within the cell and to look at what the cells are made of.
Now, what are the advantages of using a synchrotron, and how do you transfer the information you find here back into the lab to look into it further?
The advantage of using a synchrotron is that gives you a very bright infrared beam, which allows you to study not just one single cell, but also areas within the cell. This is very difficult to do with what we call benchtop spectrometers, instruments that you can have in a pathology lab, for instance. So the idea is to find out these differences using the synchrotron, and once we've got these differences, these markers, then we can use benchtop spectrometers that could be placed at pathology departments in hospitals, then to characterize these cells that are considered abnormal but not cancer yet.
What have you managed to see so far then? So what are the key differences between a cancerous cell and a normal one?
Well, this is a pilot study, so we'll still need to do a lot of work. Preliminary work which show that we are seeing differences in the protein concentration and the structure of some of the proteins and also on the DNA.
Now, you have some samples here in front of us now, so could you tell me a bit more about what's on these slides? What are we looking at?
These slides come from patients with lung cancer and contain small amounts of tissue that contain both normal cells and cancer cells. So we take these samples to the synchrotron, and then we shine the infrared light onto each individual cell, trying to find these markers.
How is this going to help the situation as a whole? So what are the current problems when it comes to lung cancer and diagnosing it?
At present, when we have got somebody from which we suspect he or she has got lung cancer, that person would have a biopsy, usually a bronchoscopy, that is, they put a tube along the nose or the mouth down to the main windpipe and try to get a piece of tissue. And then from there, the pathologist, the doctor who looks at the sample, will tell me whether it's cancer or not. However, sometimes the samples are too small or they contain a very small number of cells. And the pathologist will say, well, some cells are abnormal, but I'm not quite sure whether they are cancer. In these situations, the patients have to undergo further biopsies with all the risks and side effects for them, delaying their treatment and also increasing the costs for the NHS. So this is the problem.
So your hopeful method will enable them to look at single cells and therefore diagnose a bit earlier or a bit more specifically?
Yes, that's the idea. To be able, using an infrared light, to look at those cells that are considered abnormal, and then based on the data we obtain with infrared light to say, yes, they are cancer, so the patient can start treatment quicker.
So how much earlier do you think it might be possible then to catch the cancer?
I don't think it's a matter of catching the cancer earlier. It's a matter of helping patients to start treatment quicker. That could be 2 or 3 weeks difference. I think that's what we're aiming here, rather than catching the cancer earlier.
So you've mentioned you've got some of these slides here. So this one in particular, could you just tell me what's on here and what are the differences? At the moment, there are 3 samples really on there, I think. And what are the differences between these?
These 3 samples are 3 cuts from the same piece of tissue at different levels. And they show part of lung tissue, which is normal, and also part of lung cancer. we'll use the infrared beamline to study just one of them, and we'll look at cells that are within the normal part of the lung and within the cancer.
How would you then summarise the stage that you've currently reached with this study and what then the next few steps are hoped to be?
Well, at the moment we started this study, which is a pilot study. We try to include 50 patients, and from each patient to get a sample with cancer and a sample without cancer. Based on the data obtained on this pilot study, and if we can show the differences between lung cancer cells and normal cells using infrared beams, the next step would be to include loads of patients, and I'm talking hundreds, if not thousands of patients, that would help us to confirm that these markers indeed exist.
Would it then be possible to scale this up or look out into other types of cancer then?
Yes, we've started with lung cancer because this is the main area of my work, but I can imagine this could be extended to many other types of cancer as well.
That was Dr. Josep Soler Suso from the University of Kiel.
Did you know it took Dorothy Hodgkin 12 years to solve the structure of insulin? If she were to do this today on one of Diamond's MX beamlines, it would only take 15 minutes.
You're listening to the Diamond Light Source podcast, and this month we're entering the clinical world to find out how hospital-based clinicians are using Diamond's light source to learn more about a variety of medical issues. And still to come, we'll be finding out why metal-on-metal hip replacements could cause some side effects in patients, and how clinicians are trying to identify the possible causes. But before that, let's join Sarah Bucknell from Diamond's communications team for a roundup of the latest news and events from Diamond.
The latest news from Diamond is really exciting. We've had approval for funding for Phase 3 of Diamond Light Source, which basically means 10 more beamlines added to the facility. At the moment, we're drawing to the end of Phase 2, where we'll have 22 operational beamlines by 2012. And over the next 7 years, we're going to add 10 more beamlines, so we'll extend capabilities and increase scientific output.
Now, what makes this Phase 3? So how is it going to differ to the beamlines that are finishing off just now on Phase 2?
Well, we go out to our scientific community and we ask them what they want from Diamond. From that, we draw up proposals for these beamlines that we want to add on. The next 10 beamlines are really varied, and they'll be for a wide range of scientific disciplines. One of the beamlines will provide high-resolution 3D images of biological samples. So they'll enable scientists to look inside the cell at the scale of a few millionths of a millimetre, and that would be useful, for example, with the analysis of bone calcification or radiation damage of cells, and basically could further our knowledge of diseases and help develop new therapies. And then moving into a different area, we could also branch out into food research, allowing scientists to understand the viscosity of substances.
So that will result in an even wider range of science taking place here at Diamond. Speaking of which, there have been some recent papers published in a key journal, the journal Science, recently. What were these about?
Well, there was one recently which will help with the development of industrial catalysts. Researchers from universities of Manchester and Cardiff have succeeded in engineering crystals that are able to maintain a porous structure, which means they can carry out chemical reactions inside that crystal structure. Previously, the pores have been maintained by solvents, and as soon as you wash those solvents away, the structure collapses.
So they're focusing on just making more stable catalysts, and they've been getting their ideas from nature, from enzymes, in fact.
Yes, they've taken their initiative from enzymes, which are basically nature's catalysts. And the design of the new crystal is such that it can exist happily in water-based environments. So we're talking about industrial catalysts that don't need to exist in a chemical environment, and that are able to trap gas molecules and basically carry out those chemical reactions.
Well, it's always important to improve the catalysts used in our industries. But now moving away from chemistry and onto biology, some key findings about a specific protein have been found out.
Yes, this is a really recent paper in Science. Researchers from the universities of Leeds, Oxford and Imperial College London have determined the 3D structure of a single transporter protein in basically all its three main structural states. This protein is responsible for getting essential chemicals into the cell, and it sits within the membrane of a cell. Some of the scientists used the membrane protein laboratory that's based at Diamond to map out the structure of this protein.
So how has the protein been found to work then?
Well, this group have been looking at this for 10 years. They mapped out the first 2 states of the protein and they published those results a couple of years ago. And they finally completed the puzzle by publishing the 3rd state. And what it is, if you can imagine this protein, as I say, is sitting in the membrane of a cell. A cavity is open facing away from the cell where the chemicals can go in to that cavity. It then locks up, so this is the 2nd state, and then it opens up a cavity inside the cell which releases the chemicals, so that is the 3rd state. And so now they've got a really detailed idea of how the chemicals are basically transported into the cell.
How can this information be used? What are potential applications for it?
Well, one of their unexpected findings is that this is the case for many transporter proteins, whereas they thought it was more of a standalone. Now that they've got detailed knowledge of the mechanism, they can hopefully look at new drug developments. Altering the delivery of compounds into a cell is a potential benefit for treating illnesses. For example, if there are conditions where certain chemicals are lacking and need a boost, they can use this mechanism to help. One example would be diabetes, if there's a need for more glucose, or if somebody suffering from depression would need more serotonin.
Well, some pretty important findings there, but now stepping away from the research, you've got quite a few exciting things coming up at the Royal Society this summer.
Yes, so we're taking part in the Royal Society Summer Science Exhibition. This time round it's a bit of a bigger deal because the Royal Society is celebrating their 350th anniversary. So the exhibition is 10 days long, from the 25th of June to the 4th of July, and it's going to be at London's South Bank Centre. And it's basically a gathering of lots of interesting, exciting science that's going on around the country, and Diamond will be taking part in that. We'll have some researchers there who are using Diamond to study Alzheimer's. There's a chemist who's doing research into more efficient hydrogen storage, and also a journey into atomic and molecular research under high pressures and temperatures. So there's lots coming up, and it promises to be a really good event. So people just need to keep an eye on our website to find out more.
Sarah Bucknell from Diamond's communications team there, who'll be back in the next edition with more news from Diamond. But in the meantime, if you'd like to find out more about the work taking place at Diamond, come along to their stand at the Royal Society Summer Exhibition in London this summer, from the 25th of June to the 4th of July. Now, moving back to the science taking place at Diamond, this month we're investigating the world of clinical science and the research taking place with clinicians across the UK. Our last feature for this month's podcast looks into the hip replacement. Thousands of hip replacements are performed in the UK each year, and scientists are constantly trying to improve the materials used to provide longer-lasting replacements with maximum bone conservation. A more recent development was the metal-on-metal hip replacement, which was also used in hip resurfacing procedures. But in some patients, these have led to adverse reactions and inflammation around the hip. Dr. Alistair Hart is an orthopaedic surgeon based at Imperial College London, and I met him at his labs at Charing Cross Hospital in London to find out more about the basics of our hip joints and what's thought to be going wrong with these replacements.
Our hip joints are ball and socket joints. The ball of the thigh bone, or femur, fits within the socket of the pelvic bone, the acetabulum. When this joint is worn out, we call that disease arthritis. The most common type of arthritis is wear and tear, or osteoarthritis.
And so what are the current options available for people with osteoarthritis or hip problems in general?
Either they take painkillers and try to live with it, or if the hip is completely worn out, they undergo hip replacement. We can offer hip replacements that are either replacing just the surfaces of the bone, as in hip resurfacing, or by removing the head of the femur or thigh bone, as well as the socket part of the pelvis, and that is a standard hip replacement.
Now, the standard hip replacement has been around for a long time, and the hip resurfacing is slightly newer technique. What are the different materials available for this hip resurfacing?
There is only one type of material that is suitable for hip resurfacing. That is cobalt and chromium alloy. It is the only material that can be made thin enough to simply replace the 3mm worn-out cartilage and still stick to the bone. It can be made to reline both sides of the joint and encourage a thin film of fluid to separate those two metal surfaces so that the wear of the surfaces is very low. The hope for hip resurfacing was that it would outlast standard total hip replacement, and that if a second hip replacement was required, then there would be more bone conserved for the second hip. Unfortunately, recent evidence points towards a higher failure rate following hip resurfacing than standard hip replacements.
And why are these thought to fail at a faster rate?
The likely mechanism of failure involves the metal-on-metal joint couple. If the two surfaces are metal against metal, then that seems to cause more problems.
Now, we're just in your lab at the moment, and you've got a few failed hip joints in front of us that have used the hip resurfacing technique. So could you just take me through this particular sample here? So you've just taken one failed hip resurfacing joint out now. So tell me a bit more about this one.
The first one is hip resurfacing, and we see that the bone appeared to have been well fixed both to the cup implant and to the head implant.
The actual movement between the two is reasonably smooth.
It does feel smooth, and in fact in the body it probably is even smoother.
It's a failed joint essentially, but it doesn't really look like it. So what would have happened here for this to have failed?
The most common pattern of failure at the moment seems to be unexplained. Patients, after only a few years of having had the new metal-on-metal hip, develop pain in the hip. Some of them develop inflammatory lumps when we perform MRI scanning. It does seem to be linked to the amount of metal that is released from the surface.
So the fact that this metal-on-metal does wear, this causes the release of nano-sized metal particles into the tissue surrounding the patients with the hip resurfacing, and this could therefore be one of the potential causes of the failure.
Yes, all hip replacements wear. In fact, a metal-on-metal hip that is well functioning releases 1 million very small nanoparticles every step, and as we walk 1 million steps per year, we release 1 trillion nanoparticles into the hip joint every year.
So that's a lot of nanoparticles being released. So what is thought to be causing the damage as a result of these particles being released?
We know that the hip implant is made of cobalt, chromium and molybdenum in the ratio 60-37. However, there are many possible culprits for the active metal species that causes the inflammation and the failure. For example, is it the metal particles of cobalt, chromium, molybdenum, or is it just the cobalt or the chromium or the molybdenum, and which type of cobalt, chromium or molybdenum? And do these metal elements recombine into a larger molecule that is the active species?
That's a lot of questions that need to be answered, essentially. So how are you actually setting about looking into this?
When we collected the failed hips, we also collected the tissue surrounding the failed hips. We examined this tissue at the Diamond Light Source for both physical and chemical characteristics. We use a high-intensity X-ray beam at the I-18 microfocus beamline at Diamond. It is probably the only method of determining exactly the type of chemical in the tissues.
Now, you have various images and results in your office of what you've actually managed to find then using these techniques. So I think we're now heading back to your office in order to see these.
Yes, let's go and look at the chemical maps and the spectroscopy that's shown us exactly the type of metal in the tissues.
So we're now inside your office, and you've got various images and graphs set up in front of us. So what have you been able to find out so far about where these nanoparticles of metal are migrating to, or where they're located?
What we first did was use the Diamond X-ray beam to map the metal within the tissues of over 30 different tissue specimens. We found that these contained mainly chromium. We did not find many areas that had cobalt, chromium and molybdenum in. The majority of the areas showed just chromium. In other words, the alloy had changed from the hip replacement to how the cells and the tissues were incorporating it.
So what kind of chromium did you find?
One of the abilities of the Diamond Light Source was that it was able to tell us exactly what valence or oxidation state the chromium was in. We found that it was in the chromium 3 valence state. There are many types of chromium, including chromium 6, which is a cancer-causing chromium. We did not find the chromium 6. This does not exclude that it's never present, but it does fit with the clinical data, which suggests that there aren't cancers around these hips, but there are inflammatory lumps. We went on to show that the chromium-3 is actually present as chromium-3 phosphate.
What's the next step from here? What can you also do knowing this to try and deal with the situation?
Now we have clues as to what agents we can give to cells in a test tube in a controlled experimental environment to see if we can recreate the inflammation that occurs within a human. It may not necessarily be the chromium phosphate as the active agent. We do now know the types of cobalt, molybdenum and chromium that we should test, and then what we want to do is then take those cells that have been treated experimentally with the active agent and put them back into the diamond light synchrotron to see if we can produce the same images from patients.
But now, having done all of this then, what is thought to be the potential solution here?
I'm not sure we actually know the answer to that. The orthopaedic companies are trying many different avenues, and that probably illustrates the confusion. So, for example, it's possible to have a hip replacement that's made of metal on metal, ceramic on ceramic, metal on plastic, but it's also possible to have them made of a ceramic on metal combination. There are polyurethane bearing surfaces being used, and so it appears that we are still quite a long way from a well-accepted everlasting hip. It is probably sensible to develop better biocompatibility tests, that is, tests that are more realistic. So instead of stifling innovation, we can actually accelerate these new types of bearings, these new types of hips, but in a way that's safer.
Alistair Hart from Imperial College London. Now that's it for this edition of the Diamond Podcast, but do join us again in July when we'll be bringing you the highlights from the Royal Society Summer Exhibition, with a focus on the research being showcased there by Diamond. In the meantime, if you have any questions about Diamond or the research taking place there, the email address is podcast@diamond.ac.uk. Or you can listen to previous editions of this programme online at diamond.ac.uk/podcast or nakedscientist.com/diamond. You can also subscribe on iTunes. Thank you to Anna David, Joseph Sule-Suso, Alistair Hart and Sarah Bucknell. I'm Mira Senthilingam. Thank you for listening and see you next month.
The Diamond Podcast is brought to you by Diamond Light Source and produced by thenakedscientists.com. There's more information on our website at diamond.ac.uk/podcast.